Nutrition is of two types. That is autotrophic mode of nutrition and heterotrophic mode of nutrition. Autotrophic mode of nutrition is also basically of two types, that is the photoautotroph and chemoautotroph. And today we'll have to discuss about the photoautotroph, which is made by the combination of two words. One is the photo and another is the synthesis. Photo means production of the food in presence of light. And the light, the sunlight, the PAR, P-A-R, that is called photosynthetically active radiation, which will participate in the production of the food in photosynthesis. This photosynthesis can be studied under two subheadings. One is the light dependent reaction and another is the light independent reaction. Light independent reaction means the reaction which will take place either in light or in absence of light. So it is also called dark reaction. And light dependent reaction is the production of the which is helpful energy, which is helpful in the formation of the helpful in the dark reaction. This light reaction and dark reaction will take place on mainly in the chloroplast, and the chloroplast has a small parts that is called thylakoids. This is called thylakoid, and the whole of the thylakoids is called grana, which is connected together by intergranal lamella. And this thylakoid have these pigments. This pigments is mainly responsible for the photosynthetic reactions. Now, light reaction can be studied. Light reaction will be studied under two subheadings. One is the photo excitation of chlorophyll and photolysis of water. We will discuss both these subheadings in separate way. What do you mean by this photo excitation of chlorophyll? There are two centers for the photo excitation. One is called PS2. That is called photo system 2 and having 680. And second one is the PS1, that is 700. In this reaction center, chlorophyll is present. As you know that in addition of chlorophyll, so many other pigments are also present, and these are called accessory pigments. These pigments and this system absorb solar radiation, and by this particular solar radiations, chlorophyll release electron, that is a pair of electron at the electron acceptor center. This electron acceptor center ultimately release two electrons, firstly to the PQ, then to the cytochrome, then to the plastocyanine. Two electrons, a pair of electrons will be here. Here, a photophosphorylation means of phosphorylation. It means addition of phosphate in presence of photo light. Here ADP is converted into ATP by addition of one inorganic phosphate. And in this position, the ADP convert into ATP. Now, this electron move at this particular PS1. Here also so many accessory pigments are present. The accessory pigments receive the solar radiation, and the same function is here in the excitation of the chlorophyll to release a pair of electron and received by the electron receptor center. A pair of electron will be moved to the FD, that is called ferrodoxin, and here another energy rich compound that is called NADP2 NADPH is H positive is produced. This is the two energy rich compound will be produced by this particular photo excitation of the chlorophyll. Now, what is the function of this particular photolysis of water? You know that this chlorophyll is a chemical which will be after excitation, they will be not in the ground state. So to convert this excited chlorophyll into ground state, it is very much essential they will have a need of electron. This H2O by solar radiation convert into H positive and OH negative. This 4 OH negative convert into 2 H2O plus O2 plus 2 electron, and this electron will go inside it to convert the excited chlorophyll into ground state for the further excitation. This is called light reaction, and by this particular light reaction the ATP and NADPH2 is produced, and that is helpful in the dark reaction. In this particular first lesson we will study about the light reaction. In continuation of the light reaction, just we have studied in the previous lecture that in light reaction we produce assimilatory power. This light reaction produce assimilatory power, that is in form of ADP and NADPH2. This is the assimilatory power, and this true power is utilized in the dark reaction for the production of the carbohydrate, that is sucrose, fructose or glucose. How this particular ATP and NADPH2 is utilized in dark reaction and which chemical participate in it is just studied by we people. Now, this dark reaction can be studied in 3 steps. Number 1, carboxylation. Number 2, reduction. And number 3, regeneration of RUBP. Now RUBP, it is very much essential to understand what is the meaning of this RUBP. RUBP, ribulose 1,5 by phosphate or ribulose biphosphate. As you know that ribulose means OAC. OAC indicate that it is a carbohydrate component, and ribose indicate that it has 5 carbon. So this ribulose 1,5-biphosphate is made up of 5 carbon, and OAC indicate that it is a type of carbohydrate. Biphosphate, it means it has 2 phosphate molecules. During carboxylation, what happens? The one molecule of this ribulose 1,5-biphosphate receive one molecule of carbon dioxide, but it is possible when rubisco is present. Now, what do you mean by rubisco? Rubisco means the CO name, this particular 2 is the just this particular CO indicate that what is the function of this rubisco. Rubisco means ribulose biphosphate carboxylase oxygenase. This is rubisco. It means this is responsible for carboxylation as well as oxygenation. But in this particular C3 cycle, it is only functional in carboxylation, not in the oxygenation. Because it has been seen that in C3 plant, photo respiration also take place, and during this photorespiration this oxygenation will take place, and where there the rubisco is also involved, so their name is carboxylase oxygenase. But in C3 cycle only carboxylation will take place. Okay, now after getting this carbon dioxide, it converts into PGA. Now, what is the full form of PGA? PGA. Phosphoglyceric acid. This is the phosphoglyceric acid, and it has 3 carbon. 5 carbon from here, 1 carbon from here, and this compound with 3 carbon, it means the 2 molecule of PGA is formed. From RUBP, 1 molecule of RUBP, 2 molecule of PGA is formed. As you know that phosphoglyceric acid, and this phosphoglyceric acid, after utilizing 2-ATP to 2-ADP, here 2 molecule of inorganic phosphate is evolved, and it form 1,3-diphosphoglyseric acid. It means 2 molecule of ATP is utilized here in the conversion of the PGA to 1,3-diphosphoglyceric acid, and this ATP is produced from the light reaction. And this whole of the part, this part is called carboxylation. This part is called carboxylation. Now, come to the reduction. As you know that when acids convert into aldehyde, then we can say that the reduction has been takes place. What happens here? The 1,3-diphosphoglyceric acid. 1,3-diphosphoglyceric acid. Now, after releasing 1 molecule of the inorganic phosphate, convert into one phosphate containing substance. But here what happens? NADH plus H positive convert into NADH. And one molecule of inorganic phosphate is also being evolved during this particular process, and this is called reduction. And the product is 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde, and this is called reduction. Why? Because here this protons are being attached, and one molecule of the phosphate is evolved by this particular process, and it has also 3 carbon. Now here 3 carbon, here is 3 carbon, here is 3 carbon. It means 2 molecules of 3 carbons are also here. Now, as you know that if RUBP is utilized by this particular process, then the RUBP is not still in reserve, so the process will be stopped. So it is very much essential that the regeneration of RUBP should be take place in this each and every. Now, what happens here? The regeneration of RUBP will be takes place. But in addition to that, one molecule of C6H12O6 is also being evolved, and that is called fructose. Here you will see that the 3 carbon convert into 6 carbon. In process of glycolysis, what happens? The pyruvic acid having 3 carbon, will be produced from glucose. Pyruvic acid having 3 carbon will be produced from glucose, and glucose has 6 carbon. And this process is called glycolysis. So here we see that the 6 carbon is going to convert into 3 carbon, but here the 3 carbon is going to convert into 6 carbon. So it is called glycolytic reversal. Now, this process is called reduction, and this process is called regeneration of RUBP. Now in this process you will see that the RUBP is involved in this particular reaction, and RUBP is reformed by this particular reaction, so it will be a continuous process. And now, how many ATP and NADP is involved in the production of the one molecule of the glucose? Here, what happens? The 6 molecule of CO2, 6 molecule of RUBP. 6 molecule of CO2 and 6 molecule of RUBP. You have seen that the 1 molecule of RUBP, when receive 1 molecule of carbon dioxide, then they utilize 2 molecule of ATP. And 6 molecule, it means into 6. It means what happens here? The 12 molecule of ATP is being utilized, and 6 molecule of NADPHT is also being utilized, and one step is also here. Now you will see that the 3-phosphoglyceraldehyde converted to RUBP. There is a number of reactions here. Not directly it will convert into having C4 carbon, C5 carbon, C7 carbon, like that. And the 1 molecule of AT is also being utilized here. So the total ATP consumption is 18 and total NADP consumption is 6. And in in this process, the one molecule of fructose will be evolved. So during photosynthesis, you will see that the 6CO2 when reacts with the 12H2O, then 6C6H12O6 plus 6H2O plus 6O2 plus energy is being evolved, is being formed. And we have discussed previously that the O2 and H2O is produced in light reaction, energy is produced in light reaction, and C6H12O6 is produced in the dark reaction. So this is the whole about the photosynthesis.